Hello students, good morning to all. In earlier class we discussed about genetic disorders. In the genetic disorders, Mendelian disorders. In Mendelian disorders, autosomal dominant disorders we discussed. In the gene disorders or Mendelian disorders, autosomal dominant disorders we discussed. Today's class we are going to discuss about autosomal recessive type of disorders. So in earlier class we discussed autosomal dominant disorders along with the pedigree analysis. Today, today's session we are going to discuss about autosomal recessive disorders. Pedigree analysis we will discuss in next class. Okay. So autosomal recessive disorders. One of the example is sickle cell anemia. For autosomal recessive disorders, one of the example is sickle cell anemia. So today class I am doing throughout the NCERT lines, line to line NCERT, because so whatever lines given in NCERT, all the points are very most important, right? So while you writing the notes, so point wise write it from the NCERT textbook. Okay, right. So sickle cell anemia. Sickle cell anemia is the example for autosomal recessive disorder. So in the Mendelian disorders or gene disorders, sickle cell anemia is one of the example for autosomal recessive disorder. So first point, this is an autosome linked recessive trait that can be transmitted from parents to the offspring when both the parents are carrier for the gene or heterozygous. So this is the example for autosomal recessive disorder, and it can be transmitted from parents to the offspring. So whenever parents are suffering with sickle cell anemia, then only it is transferred to the offsprings also. So at parents are what condition? Parents are parents are carriers for the gene. Parents are carriers for the gene means parents are heterozygous. Earlier we discussed autosomal dominant. Autosomal dominant means in case one parent affected, another parent normal, then affected children are there. Some of the children are affected. Okay, right. So that is dominant means what? Only single gene also presented. It is phenotypically expressive, but it is autosomal recessive type of disorder. So autosomal recessive type of disorders, for recessive, how many genes are required for phenotypic expression? Two genes are required. So two genes are presented in only one parent. One parent affected, another parent normal. It is possible for expression of disease in the children. It is not possible. So that only. So both parents are carriers means both are heterozygous parents. One parent is carrier, another parent also carrier means one parent having one recessive gene, another parent having one recessive gene. So both recessive genes phenotypically expressive, then only it is the disease can be expressive in affected children's in next generation. You understood or not, right? So this is autosomal linked recessive trait that can be transmitted from parents to the offspring when both the parents are carrier for the gene or heterozygous. Okay, right. Next point, the disease is controlled by a single pair of allele. The disease is controlled by single pair of allele. So alleles are HBA and HBS. HBA and HBS. There are two types of the alleles. So out of the next point, out of the three possible genotypes of out of three possible genotypes only, homozygous individuals for HBS show diseased phenotype. So here see, HBA means normal, HBS means sickle cell anemia. Okay, so HBA, how many genotypes are possible? Three types of genotypes are possible. What is that? HBA, HBS, HBS, HBS and HBA, HBA, right? So in this condition, which are disease individual, so it's HBS and HBS only, because because what is the reason? Because both are recessive genes, phenotypical expressive recessive genes only affected individuals. So out of three possible genotypes, only homozygous individuals for HBS show the diseased phenotype, right? Okay, heterozygous HBS individuals appear apparently unaffected, but they are the carrier for the disease, as there is 50% probability of transmission of mutant gene to the progeny. So HBS means affected individuals. HBS means what? HBS. These two, the HBS are individuals are unaffected. Why? Only one HBS is presented, okay, but these are carriers. For example, one parent is HBS, another parent is HBS. In progenies, how much chances are there? That given, go through the point. Heterozygous HBS individuals appear apparently unaffected, but they are carrier of the disease, as there is 50% probability of transmission of the mutant gene to the progeny, right? So 50% of transmission of the disease is there. Suppose 4 members are there, so 2 members are going to be affected in the individuals. In what case? In case both parents are heterozygous condition only, okay, right? Only one parent heterozygous condition, another parent normal means it will not come, okay, right? Thus exhibiting sickle cell trait, thus exhibiting sickle cell trait, okay, right. Okay, here the point is heterozygous HBS individuals appear apparently unaffected, but they are carrier of the disease, as there is 50% probability of transmission of the mutant gene to the progeny, right? 4 members are there, 2 are affected, thus exhibiting sickle cell trait. So which people, what type of the people or individuals are affected individuals? In case both parents are carriers only in autosomal recessive disorder, right? One parent is HBS, one parent is HBS, then only it is possible for expression of a defect in the individuals. Understood or not, right? Okay, next one. So what is the reason for this disorder? So what is the major reason for the disorder? So why this type of sickle cell anemia is formed? The reason is the defect is caused by the substitution of glutamic acid by valine at the sixth position of beta globin chain of the hemoglobin molecule. So the hemoglobin molecule, which is made up of with there is different types of amino acids, right? So by the amino acid sequence, the hemoglobin molecule is formed, so protein formed by different types of amino acids. So this defect is caused by the substitution of glutamic acid by valine. So glutamic acid is replaced by valine. So whatever the position of glutamic acid is replaced by the valine, at which position? In the sixth position of beta globin chain of hemoglobin molecule. In the hemoglobin molecule, in the beta globin chain, so the sixth position glutamic acid, which is substituted by valine. In the place of glutamic acid, what is substituted? Valine is substituted. So in one point only the amino acid is going to be changed. So this is the example for sickle cell anemia. Is the example for what? Sickle cell anemia is the example for point mutation. Earlier we discussed in mutation topic, right? So first point, the defect is caused by the substitution of glutamic acid by valine. Glutamic acid is replaced by the valine at the which position? At the sixth position of beta globin chain of the hemoglobin molecule. Okay, right. Next point, the substitution of amino acid in the globin protein results due to the single base substitution at the sixth codon of beta globin gene from GAG to GUZ. So whenever the glutamic acid replaced by the valine, so what happens? The substitution of amino acid in the globin protein results due to the single base substitution at the sixth codon of beta globin gene, that is GAG2 GUG. The gene sequence is changed, GAG2 GUG. So what happens? Because of only one amino acid changed, glutamic acid replaced by the valine, so that only the gene codon changed, GAG to GUG. Instead of GAG, the GUG is formed, right? The next point, the mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of RBC from biconcave disc to elongated sickle cell like structure. Okay, what is the point? The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of RBC. Sickle cell anemia. What is the shape of RBC? Generally biconcave shape. The biconcave shape RBC is changed into what? Sickle cell. The shape of RBC is changed. In the diagram given, clearly you can observe here. In the diagram given, first normal hemoglobin gene, that is biconcave shape, and whenever sickle cell gene, that is sickle cell shape, the RBC shape of the RBC is changed. So how the shape of the RBC is changed, that one given. The mutant hemoglobin molecule undergoes polymerization and under low oxygen tension, causing the change in the shape of RBC from biconcave disc to elongated sickle like structure, right? So what is the reason for this? Because of the sixth in the beta globin chain of a hemoglobin molecule, the sixth position amino acid, glutamic acid, which is replaced by valine. So whenever glutamic acid replaced by the valine, and the gene codon is changed from GAG to GUG. Whenever GAG to GUG changed, then the polymerization takes place, and in the low oxygen tension, the shape of RBC changes from biconcave shape to sickle cell shape. So whenever sickle cell shape changes, RBC shape is changed, then what happens? So we discussed it in respiration chapter. So transportation of the oxygen majorly takes place through the blood plasma and RBC only, right? So whenever RBC shape changed, then the percentage of oxyhemoglobin is decreases. The percentage of oxyhemoglobin is decreases. So oxygen transportation is decreases to the all the body parts. That is sickle cell anemia, right? Understood? Once again, I am revising from starting. Okay. So sickle cell anemia, this is an autosome linked recessive trait that can be transmitted from the parents to the offspring when both the parents are carrier for the gene or heterozygous. Okay, right. So what is the example? Sickle cell anemia is an example for what? Autosome linked recessive trait. Okay, right. Next point, the disease is in what case is it is going to be spreads to the, it is going to be carry forward to the individuals, both parents are heterozygous condition only. Okay, right. In normal case it is not possible, only one parent affected parent is there and second one is normal, it is also not possible. One is affected, another one is carrier, yes, it is possible. Why? Because it is recessive, two recessive genes are required for expressive, phenotypical expressive. Okay, next point, the disease is controlled by a single pair of allele HBA and HBS. So HbA and HbS controls the this disease, and whatever the alleles are HbA and HbS, and out of three possible genotypes, only homozygous individuals for HbS show diseased phenotype, right? So here what HbS and HbS only there is a chance for expression of disease. When HbS is there, then there is no expression of disease because of it is recessive, and HbS is shows the diseased phenotype. Means what? Both the parents are having HbS gene, either carrier or affected. You understood or not? Okay, right. Next point. Heterozygous HbS individuals appear apparently unaffected, but they are carrier of the disease, as there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting a sickle cell trait. Okay, so here what happens? HbS, so those are HbS are what? Those are carriers only. Why? Because it is not going to be unaffected. HbS condition is unaffected, but they are carrier of the disease, and there is 50% of probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. Okay, right. So what is the reason for the disease? Because of the defect is caused by the substitution of glutamic acid by valine. At which position? At the sixth position. In what? In the beta globin chain of hemoglobin molecule. Glutamic acid in the sixth position. Glutamic acid is replaced by the valine in the beta globin chain of hemoglobin molecule. So by that what happens? The substitution of amino acid in the globin protein results due to the single base substitution at the sixth codon of beta globin gene from GAG to GUG. So whenever one place only there is a base pair of DNA is changed, so that only it is the best example for a classical example for point mutation. In single point only glutamic acid replaced by valine, and remaining everything should not change. Okay, so it leads to the gene codon formation. Actual gene codon formation is GUG, but glutamic acid replaced with the valine, so that only it is formed as converted into GUG. GAG to GUG, glutamic acid replaced by valine. The 2 points you should remember. You can observe in the diagram also, NCERT diagram also. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of RBC from biconcave disc to elongated sickle-like structure. So whenever the gene codon is changed, GAG to GUG, then the shape of RBC changes from where to where? From biconcave shape, normal shape of RBC biconcave, it changes into sickle cell shape. Whenever the RBC shape is changes, means then oxygen binding affinity with the hemoglobin is going to be decreases. So less oxyhemoglobin is formed. Understood or not? Right? Very most important for PU purpose and as well as objective purpose also. Okay. Once go through the NCERT lines and make the points from the NCERT textbook only. Okay. Once go through the diagram also. And the remaining disorders and this sickle cell anemia pedigree analysis we will discuss in next class. After this clear? Thank you very much. Bye bye.